Hi everyone! On this video, I will talk about a paper from the National Institute of Mental Health in Maryland. This work was performed by Monica Koenig et al. And it's about pain responses, anxiety, and aggression in mice deficient in pre-proenkephalin. First, let's refresh our memories about endogenous opioids. They are substances primarily produced in the brain, but they are also found in several other areas of the central nervous system, including the spinal cord, such as the dorsal horn. They have profound analgesic effects, and they are vital in regulating physiological functions, including pain perception, analgesia, responses to stress, aggression, and dominance. Enkephalins are endogenous opioid peptides that are derived from a pre-proenkephalin precursor protein and can act as neuromodulators and neurotransmitters. Encephalinergic neurons are present in many regions that mediate pain responses, including the mesencephalic central gray matter and the substantia gelatinosa in the spinal cord. But how do they do it? Well, when a primary nociceptive fiber is stimulated, it releases glutamate, which is an excitatory neurotransmitter. It is released into the postsynaptic terminal of a spinal sensory neuron. Nonetheless, an encephalinergic interneuron is also present, causing inhibition by releasing enkephalins into both the pre- and postsynaptic terminals. Therefore, it counteracts the excitatory input and decreases the signal that we will later interpret as less painful. With regards to anxiety and aggression, enkephalins also have a direct role in modulating emotional behavior by their widespread distribution throughout the limbic system, including the amygdala, the cingulate and entorhinal cortex, the septum, and the hypothalamus. The research in this article is mainly a proof of concept. Do enkephalin-deficient mice display different levels of pain perception and analgesia? And also, do they have different responses to aggression and dominance? In order to validate their hypothesis, multiple behavior experiments were performed. The first one involves genetic engineering to generate enkephalin-deficient mice. A vector containing a replacement sequence for the enkephalin coding section of the proenkephalin gene was designed and then transfected into blastocysts. Later, these were injected into mice to generate chimeric mice, allowing the mutation to be transmitted to progeny. The rest of the experiments assess behavior on these mice. The first one is the tail flick assay. For this assay, mice received foot shocks of 1 second duration at 5 seconds interval. Foot shocks are known to induce an opioid-dependent form of analgesia at the spinal level. Then, forced swimming experiments were performed to induce stress. Mice had to swim in a 4-liter glass beaker, and it is known that forced swimming induces analgesia, and it can be mediated by opioid systems, for example, if the water is around 34 Celsius degrees, or by non-opioid systems, depending on the water temperature. After the forced swim test, they were placed on the hot plate heated to 55 Celsius degrees during 240 seconds, and the jump latencies were recorded to measure stress-induced analgesia. The formalin test is a measure of inescapable pain. For this test, mice were injected with formalin under the dorsal surface of the right hind paw, and paw licking and lifting behaviors were recorded as normal pain responses. An O maze consists of 2 open and 2 enclosed sectors elevated 40 centimeters above the ground, and the time spent by the mouse in each sector is measured. The avoidance of open parts of such runways is indicative of anxiety. Anxiety measurement consisted in open field tests, where a 60 by 60 centimeters floor area was divided into 3 by 3 squares, and the behavior of the mouse was recorded during 15 minutes. So, if the mice explored or crossed to another square more frequently, they were less anxious. Lastly, aggressiveness was measured through a resident intruder experiment. The mice were housed individually for 4 weeks, and when another mouse was placed in the cage, they would count the number of attacks and how long they lasted as a reference of the intensity of fighting. Let's move on to the research findings. The first set of results are related to the targeted mutagenesis of the expression analysis of enkephalin in genetically engineered mice. This first figure mainly validates the creation of the enkephalin deficient mice. In panel A, on the first line, you can see the plasmid construct that substituted the coding region for the enkephalin gene. On the last line, we can see that when substituted, then we end up with an enkephalin knockout locus. B and C are images of northern blots. This molecular technique allows to study gene expression by detecting RNA where the bands indicate the presence of enkephalin. On B, we see that enkephalin was reduced by about 50% in heterozygous mice, and on C, on the last two columns, there is an absence of enkephalin in homozygous knockout mice. Panel D shows that only the enkephalin levels were reduced by 44% in the knockout mice, and no differences were found in the expression levels of the other endogenous peptides, like dynorphin and endorphin, between genotypes. After validating that the expression of enkephalin was actually knocked out, these new mice were used for the behavior test. Figure 2 shows the results for the tail flick method to assess pain sensitivity and stress-induced analgesia mediated at the spinal level. As you can see on 2A, the pre-stress latencies were identical on wild-type and knockout animals, meaning that the knockout animals exhibit a normal pain response. After receiving the foot shocks, both latencies increased, indicating that similar levels of analgesia were induced. In contrast, during the hot play assay on Figure 2b, the knockout animals showed a significantly shorter jump latency, indicating that the supraspinal pain threshold is lower in knockout mice and evidentiating that enkephalins are involved in the modulation of pain responses. Graph C corresponds to the forced swimming test at 34 Celsius degrees to measure induced analgesia. Here, both genotypes had similar results, indicating that this form of analgesia, although it's opioidergic in nature, is not mediated by enkephalins. Interestingly, if you look at Figure 2D, they once again did a forced swim test, but this time at 4 Celsius degrees, and again, there was no significant change, indicating that enkephalins are not involved in stress-induced analgesia. Figure 2e shows the results for the formalin injection test. This graph indicates that behavioral alterations are a direct consequence of lack of opioid peptides, since the knockout mice showed increased paw lifting and licking. This in comparison to the wild-type mice that received the injection of naloxone, an opioid antagonist, where similar percentage on pain response was shown. Figure 3 shows the results that assess the locomotor activity, where knockout animals displayed significantly reduced levels of horizontal activity. Panel B and C reflect increased apprehension or anxiety, as the knockout mice prefer to stay on the peripheral areas of an open field. They also spent less time in the open sectors of the O maze, because being on these open spaces makes them feel exposed and triggers their anxiety, so they avoid it by moving to the enclosed areas. Finally, the aggressive behavior of the knockout mice was assessed during the resident intruder experiment. Figure 4 shows that knockout mice were often aggressive, especially when housed individually. As previously explained, these mice were kept in a cage by themselves and a new mouse was placed inside the cage. Here we see that they were immediately intolerant to intruders as they attacked them on a higher frequency and during a longer period of time. In other words, they were simply more aggressive. To recap, opiates have profound analgesic effects. It has been suggested that the endogenous opioid system might be important in modulating reaction to painful stimuli in situations where environmental danger is perceived, such as stress-induced analgesia. Specifically, enkephalins are part of the endogenous opioids, and they are responsible for mainly modulating our pain perception and emotional behavior via inhibition. Through the results of this variety of tests, together, they prove that the behavioral changes produced by the preproenkephalin mutation can be described as an exaggerated response to painful or threatening environmental stimuli. Pain is a highly subjective sensation. Although nociception at the spinal level is not affected by the pre-proencephalin mutation, the perception of pain seems to be different in encephalin knockout mice, as indicated by altered supraspinal pain responses. Interestingly, further research in humans shows that opiates do not change nociceptive thresholds, but rather reduce the subjective feeling of pain. Thus, encephalin knockout mice may provide an animal model for future investigation of neuronal pathways and processes that mediate the experience of pain. Thanks for watching!